Hi everybody. So in this video we're going to be talking about antigen presentation and specifically how we are going to incorporate antigens on the surface of Mhc Class 1 and Mhc Class 2. So we have two pathways that we're going to be talking about. The first one is how we're going to be incorporating antigen on the surface of Mhc Class 2 molecules. And the second one is how we're going to be incorporating antigen on the surface of Mhc Class 1 molecules. And in the case of Mhc Class 2, we're going to be mostly talking about exogenous antigen. Whereas in the case of Mhc1, we're going to be talking about mostly endogenous. So just what is the purpose of Mhc Class 1, Mhc Class 2? Well, they're located on different cells. So in the case of Mhc Class 2, this is going to be on the surface of specifically antigen-presenting cells. So we're thinking macrophages, dendritic cells, and B cells. Whereas in the case of Mhc Class 1, we're really talking about any nucleated cells. So all nucleated cells. And this includes antigen-presenting cells, but mostly just all cells that you can pretty much think of besides like red blood cells and some other specific non-nucleated cells. Pretty much all cells in your body are going to have Mhc Class 1. And Mhc Class 1 is what is going to interact with CD8, cytotoxic lymphocytes, and Mhc Class 2 is what's going to interact with CD4, T helper cells. How do each of these pathways work? So we're going to start with Mhc Class 2, the pathway here that we have going at the top with exogenous antigen. So we have some sort of exogenous protein source that's coming from outside the cell, right? Because that's what exogenous means. So that protein source is going to come and enter the antigen presenting cell. So this is either a macrophage, dendritic cell, or B cell that we're talking about. It's going to enter and it's going to go straight to the lysosome. And that makes sense, right? Because lysosomes are responsible for processing exogenous protein sources. So nothing new here. And then once it goes through the lysosome, it's going to get broken up into smaller peptides. And then that peptide is going to be brought to the Golgi for processing. But what is it being processed with? Well, the whole purpose of it going to the Golgi is to be placed with an MHC2. So eventually we're going to get MHC2 on the surface of this antigen presenting cell, but with the peptide attached as well. So in order for us to process it with the MHC2, we have to actually have an MHC2, right? So we're going to produce an MHC2, and the way we do that is with our endoplasmic reticulum. So we're going to pretty much produce this receptor, which is a protein, the same way we produce any other proteins, which is through ribosomes and the endoplasmic reticulum. So we're going to produce this MHC2. And on this MHC2, we have something called invariable chain. And what this invariable chain is, it's actually just like a placeholder for this peptide that we're eventually going to put in its place. But since we're not putting on the peptide until we get that MHC, or since really that we have to bring this MHC to the Golgi before we are attaching the peptide, we need something to as a whole placeholder so that way that MHC doesn't grab onto anything that it's not supposed to. So the MHC2 with the invariable chain is going to travel to the Golgi apparatus where it's going to meet the peptide, and now we are going to kick off that invariable chain. And be able to present that MHC Class 2 with the peptide on the surface of the cell. Okay, and the endogenous pathway is pretty similar. One more thing I do want to say about the exogenous pathway is that we have something called interferon gamma, which is able to upregulate the lysosomal activity. So it's just something that we use whenever the cell, it's something that antigen presenting cells are able to utilize in order to increase the efficiency of the lysosome. And it also causes increased production of Mhc class 2. So we can go ahead and put that here as well. Okay, so endogenous pathway, pretty similar. There's a couple of key differences, which I'm going to point out. So endogenous antigen is obviously not coming from the extracellular side, it's coming from inside the cell, right? So whether this is just, any cell in the body can do this. So antigen presenting cells can do this, but also, T helper cells, like you name a cell, it can do it. So the endogenous source here is going to be going to the proteasome, which is no surprise because we previously learned that endogenous protein sources are broken apart using the proteasome. So from the proteasome, it's going to get broken up into smaller peptides. So pretty similar so far. Now the key difference here is that instead of the peptide going straight to the Golgi, it's going to go to the endoplasmic reticulum. And in the ER, we are having a similar process, right? We have our MHC, in this case, the MHC class 1. And since that peptide is able to be brought directly to the endoplasmic reticulum, we can actually put the peptide on the MHC as it's being processed. So we don't need an invariable chain. We don't need a placeholder since we're putting it directly on. And in order for that peptide to be carried directly to the endoplasmic reticulum, we have to have a protein that's called TAP, T-A-P. And then after that protein is processed in the endoplasmic reticulum, it can be brought directly to the Golgi for processing. And then that MHC1 can be presented on the surface of the cell with the peptide attached. And then one final note with the endogenous protein pathway is that the cells use an interferon called immunoproteasome. And what immunoproteasome does is it increases the speed of the proteasome, but also increases the size of peptides that it breaks up. So a similar concept to what interferon gamma does, but it is essentially making the proteasome better for the job that we're trying to get done here, which is to create a peptide to go on the MHC1. Okay, and then there's one final note, and that's a concept known as cross-presentation. Cross-presentation is essentially the ability of antigen-presenting cells to be able to take exogenous antigen and shunt it through the endogenous pathway. And this is specific to antigen-presenting cells, and it's called cross-presentation. And so what exactly would be the purpose of an antigen-presenting cell going through the endogenous route? So say we have a macrophage, and I apologize in advance because my macrophage is about to look a lot like Pac-Man. Here's our macrophage, right? And then over here we have some sort of cell. It doesn't really matter what it is. It's a cell, and it's cancerous. And in that cell, there's some sort of aberrant protein, which I've so eloquently drawn here, that if it were to be presented on Mhc class 1, it would be recognized by a cytotoxic T lymphocyte, so CD8 positive cytotoxic lymphocyte, and it would essentially cause apoptosis. Right, because that's what cytotoxic lymphocytes do. They recognize things, problems with the cell, and they can cause cell death. However, if we have Mr. Macrophage over here come along, and the macrophage actually engulfs this cell, right? So now we have the macrophage, and it's eaten the cell. and or the remnants of the cell. And now we have this aberrant protein that would have notified a cytotoxic lymphocyte that, we got cancer, okay? And now, technically, the problem is that this is exogenous protein, right? So it was endogenous when it was in this cell over here, but since it's been engulfed by this macrophage, it's technically exogenous to the macrophage. Antigen presenting cells weren't able to shunt this protein through the endogenous protein pathway. It would be presented on MHC2 because that's what happens to exogenous proteins, which is going to be a problem because our CD8 cytotoxic T cells that we need to recognize cancer aren't going to be able to recognize it in the context of an MHC class 2. They're only going to be able to recognize it in the context of an MHC class 1. So therefore, we have to have this cross-presentation ability so that this exogenous protein will be presented in the context of MHC class 1. So that way our CD8 cytotoxic lymphocytes can recognize it. Okay, guys, that's all I have for you. I hope that this video cleared up some questions that you might have had. If you have any additional questions, please feel free to leave a comment. Please like, subscribe, and I will see you guys next time.